One of the interesting things I get to do is review DNA studies. And I wanted to show a little bit of a representation of what I do in analyzing DNA data, what I'm thinking about, what the process is, the patterns I'm looking for, how I connect one spot to another spot. When we get DNA studies back from typical medical labs, there is some limits on how those labs are functioning. Typically, when a physician orders a blood study, and especially a genetic analysis, they're using it as a somebody is coming with a certain presentation. Let's say, for example, an autoimmune disorder, or they're concerned about a genetic, maybe like Ehlers-Danlos syndrome, which is hyperreflexivity. And usually they'll go to a database of labs and they review the labs available and say, we're going to collect this assay, submit it to insurance, and off it goes. Oftentimes, the assay itself it may or may not be complete or the most up to date. One of the challenges in medicine is the rapid advancement in technology and understanding the connections that all these genes make and all these genes and proteins. So a lot of times the assays that we do collect may not have the variations that we're looking at, or it may be missing a couple genes in the midst of all this. So while a lot of things can be diagnosed off of these assays, it's not necessarily a complete picture. The challenge is how do you analyze this in a more complete fashion? And there are commercial products out there. There's whole genome sequencing products made available commercially where they analyze your entire genome. They look at every DNA, every sequence, and say, well, this is what we got. Now, the challenge with this is with the whole genome sequencing is the massive amount of data that's thrown at us. And trying to interpret and sift through the weeds is very challenging. Um, there's a lot of genes that may have no influence at all. There's some that may not be complete. There may be some that are not fully interpretable. There's a lot to look at and think about when interpreting DNA, and sometimes it gets overwhelming. So people who go into like the ancestries and the 23andMe's, these studies are not whole genome sequencing. They look at specific SNPs variations that allow them to connect you genealogically with other people. To get a whole genome sequence, you have to go specifically to certain websites. There's a few companies out there. I'm not going to name any specifically just because I don't want to sound like I have a bias or anything. Um, the one that I use regularly does have a way to connect to a portal, a gene analysis portal that's available really actually publicly. I have it there on this screen, and it's gene.iobio.io is the website. And I love the name. It's just really fun to read off. But what you can do with this website is right there at the very top, you can run with demo data. And I'm actually going to click that. You can actually load your data if you have it. And of course, as genomes tend to be very large, that could take a while. So what happens is so you go to a company, you get your whole genome sequence, they put it into a huge database, they make that file available to a portal like this to analyze that DNA. And here I'm going to run with just the test DNA. And over on the left, what it does is it's analyzing each of these these genes, and it marks them with suspected variations and says this one's most likely to be pathogenic. This one seems to have an influence. This is a variation that seems to have journal articles associated with it. This one, for example, RAI1, is known pathogenic in ClinVar. ClinVar is another database that allows us to collect all of these variations and say this one has an effect, this one doesn't have an effect. So it allows us to collect more and more data together to piece together what these all mean. And sometimes we look at a variation and there's no clinical effect whatsoever. And that's not unusual. Sometimes a DNA molecule can switch from one to another and it has no relevant effect on the protein down the line. However, there's other times where a whole sequence can be taken out or there's a start stop that doesn't happen. There can be complete dysfunction of those genes. And when I look at a gene sequence or a DNA sequence and say, all right, I am interested, this person says they have gender dysphoria. I am looking for a gene that may be contributory to this. And can I find a gene and can I piece this together with other effects that make it more plausible to say this gene is the most likely culprit out of the midst of all this? So when I start out here with phenotype, and over to the left, you can click gene. One of the easiest ones to look at is factors. These are the blood clotting cascade, and factor 9 is a one that leads to hemophilia B. It's on the X chromosome, it's X-linked, and you can see it's right there with hemophilia B. This person doesn't have, or this test data doesn't have any variations on it, but you can grab a whole bunch of information on this. Now, the cool thing with this is you can take this and find journal articles and say, this is why we see what we see. And you can see that over there in the PubMed section right there, that I can now click on a link and say, hemophilia B, these are articles related to this gene sequence. That's not the only thing that I can do with this. In this, of course, only does the individual genes. I can do collections of genes. And this is where things become very interesting. So, for example, under phenotype, one of the most powerful sets that I look at is sex reversal. Now, as you've seen from my previous presentations, there's a whole bunch of genes involved in our sex development, and having this category of sex reversal makes it a very powerful tool of saying, okay, where in these DNA pathways, these gene pathways, could I expect something to happen? Do I see complete sex reversal where this person was assigned male at birth and has XX chromosomes? How would I see that? Or is this person assigned female at birth and has XY chromosomes? What would I expect to happen with these gene sequences and what pathway would I go down? Now, in here, it's only allowing me to look at 20 different genes. There's ways to look at 100 different ones. And this one shows that there's an effect on the androgen receptor. Now, I know with the androgen receptor, this is associated with androgen insensitivity syndrome. And there's partial and there's complete. Is it possible that this person may have an XY chromosome, assigned male at birth, but only has partial response to the androgen receptor? Do they have secondary characteristics? Do they have a body habit, fat distribution that's more female in origin? Or are they having breast development or gynecomastia? So those are a lot of the questions I ask about how do I look at this gene variation and clinically, can I apply it to this situation? Or if I'm lucky, maybe they have lab testing that allows me to say, all right, their testosterone or their estrogen levels are off. Maybe I need to look at the pituitary gland and say, all right, the FSH and the LH are off as well. So this allows me to create a more complete picture of an individual with this gene variation and say, what does that mean? Where can I go with this? So as I take through all of this, this is just one category. I can look up the pituitary and say analyze 100 or 20 different genes that are associated with pituitary gland development. And there's a number of them. And that's what's amazing with this is each of the structures that we have in our body, whether it be the liver, the kidneys, the brain tissue, all have these combinations of genes that turn on and off at different times in our life through gestational development, through puberty, through childhood, through adulthood. All of these things actually have timing of when they turn on and off. And that's what's really amazing and complicated about this. So I can look at others. Let's say, for example, testosterone. And I can do the same thing. I can go through each of these categories and think pituitary, I can think of sex reversal, I can think of estrogen, testosterone, the adrenal gland, what's interesting is now I want to point this out down at the bottom. Notice how it says benign and clinical var. So then VAR has analyzed this mutate or variation and said this does not appear to have a clinical effect. And there's a lot of other details on here that help to point, does this really actually have an effect on an individual? So I can take the data I have here, and then take it to a lot of the pathways that I'm familiar with, and I say, where in the sequence does this fit, and does this make sense? So here I pulled up the embryologic development of the sex organs and the gene pathways that happen in between. And I think of all these genes and I say, all right, WNT4 is a mutation or a SOX9 is a mutation. Maybe I can actually take it back and say, oh, no, no, actually, I see this person has an XY chromosome. They have an SRY gene. Why isn't it doing what it's supposed to do? What is the effect on that? Maybe the gene down the line is not working. Maybe this person, the pituitary gland is not effective. Um, so there's a lot to think about and piece together with this. Now, if I don't see a lot of things here, I can come over here and say, all right, what's happening with the pituitary gland? These are a lot of the genes that are associated with the pituitary development, which is the control center of our sexual maturity and development. And if I don't see what I need to here, I can take this a bit further and look at, well, here's the adrenal gland. Here, the adrenal gland is taking cholesterol and is turning it into all the different components that make up our body, whether it be a mineralocorticoid, a glucocorticoid, or a sex hormone. Um, so yeah, taking all this information, compiling it, and looking for patterns and asking these questions of what do I see and what don't I see. Now, what's interesting with this is a person comes to me and says, hey, I have gender dysphoria, and I find a gene here. What do I do with that? So if I have a gene mutation and I suspect there might be a clinical effect, are there other people in the world with that same variation that also has an effect? Well, that supports the hypothesis that this gene does have a clinical effect on this person, even though we may not see it directly. Example of that would be somebody with congenital hypogonadotropic hypogonadism. This is the effect on the pituitary gland. And let's say that for the most part, the pituitary gland is functional in one person, but not necessarily in another. Or we have a person who's male and a person who's female. The female individual develops primary amenorrhea, which is a lack of a menstrual cycle, because the pituitary gland is non-functional. I know then if that's the same variation that this other person has, the male has, maybe that explains why they have gender dysphoria, is the pituitary gland did not develop appropriately. And as a consequence, the masculinization of the brain or feminization of the brain never happened. So it's one of those where taking all this information, gathering information from other places allows me to compile all this together and present it to an individual. And sometimes I have to say, hey, I need more data. I need lab testing. And here's what I recommend you do going to your primary care doctor or to a endocrinologist, or if you're lucky, going into a geneticist and analyzing this. Um, Sometimes I say, yes, I see it here. I can see the effect. Sometimes what I would expect with sex hormones or sexual maturity or development has other effects as well. These genes have other clinical effects on other places. A perfect example of that would be TP53, which is a gene associated with apoptosis. It's associated with many cancers. It's associated with leukemia. Yeah, many cancers in of itself. But TP53 is associated with a lot of other conditions. And one of them is actually sex reversal in rare cases. And the primary source of that is this gene is responsible for causing cells to drop off during gestational development. And if those cells don't fall off when the right time is to do that, then they go on to continue to develop into ambiguous genitalia or other effects. Now, this same problem can cause issues with schizophrenia, this can cause depression, this can cause autism. So there's a lot of things that are associated with just one gene. And the question is how do I make it have an effect? One of the genes that's really cool about this is the um, fibroblast genes. These are genes that are responsible for getting cells to where they need to go. And there's a lot of them that are involved in sexual development. FGFR1 is just one of them. FGFR1 is also part of the pituitary pathway and getting that pituitary gland to develop the way it's supposed to and get those neurons to end up where they're supposed to. And as a consequence of that, one of the other things that's responsible for is getting the nerve cells for the nose, the olfactory bulb. So one of the consequences potentially that can happen with an individual with an FGFR1 mutation is not only effects on sex development, but anosmia, which is a lack of smell. This also has effects on bone growth. And so an individual with this mutation may also have issues with proximal bone growth, especially the long bones like the shoulders and the hips. So those are some of the things I look for is I have a gene mutation. How can I apply it clinically? Do I see the effects? Can I look at hormone levels? Can I look at FSH and LH? Can I see it all together in one complete picture? And that's what's really amazing about this. Sometimes it takes me a few days to actually analyze these DNA studies and go, okay, this is what I suspect is going on. Sometimes it's very obvious from the very beginning and it only takes a few minutes. But that's kind of my process. Hope you enjoyed it.